Am I on? Okay. Good morning, everyone. Welcome to this Saturday morning session. Glad to see everyone here. We had one speaker already mention that being up late last night at the party and trying to get up early to come give the talk is going to be a little tough, but I'm glad everyone was able to survive the party last night and then to make it here this morning for this outstanding session. We really have a great set of speakers lined up for this morning's talk. I'm Trent Spencer. I'm from Emory University.
Yeah, and my name is Jung-Hae Seo from Rice Bioengineering.
Yeah, so we're part of the new direction, the Viral Gene Transfer Vector Committee, and what we try to do each year is put together some of the state-of-the-art talks on where some of the vectors are headed, what's going to be the next generation of gene transfer vehicles. And in this talk, you're going to see that we focused on two of the leading vector systems, AAV, and then we have a lentivirus vector talk, which are among the top gene transfer vehicles being used. So we think that these are very appropriate talks at this time. But then I'm excited to hear the fourth talk as well, which is a presentation on how to start monitoring some of these gene transfer events in vivo, which is going to be a very interesting talk as well. I do have to remind everyone to silence their cell phones, and the speakers will have 20 minutes. A light will come on. It'll come on at 20 minutes. You'll have 5 minutes to start wrapping up after the yellow light comes on. So you'll have a total of 25 minutes, and then we'll have 5 minutes of talks, or if you go over, then we'll shorten the question session. So we have speakers that are well versed in staying on time, so hopefully our job's going to be easy up here. So with that, I'm going to introduce our first speaker, which I was very glad to meet this morning. Hadn't certainly followed Luke's work for several years, and it's glad to meet him, and looking forward to this talk that is entitled Ancestral AAV Capsid Reconstruction, Integrating Viral Evolution in Inform Rational AAV Design.
Thank you very much. That's what you get when you have long titles early in the morning. Thank you, everybody, for being here at this ungodly hour. Without further ado, I'll start by putting up my disclosures. Over the past 18 or so years, we've developed quite a few technologies, and they've passed through various hands, highlighted in red here, and that's the main source of these conflicts that I need you to be aware of. The ones highlighted in red are, I think, the ones most pertinent to the talk here. Before starting, I do want to acknowledge that this is actually a body of work that has emerged out of my lab over the past 6 years. I have an incredibly talented team of passionate people around some delivery questions that we're asking in the lab. I try to highlight this throughout the talk, but I can't indicate everybody's individual contribution given the limited time. But these are some of the people, these are all of my ex-members, former members, collaborators, and so forth that I am indebted to. So to kind of frame my talk, the way I look at the questions asked in this field is that we're really trying to formulate the gene as a drug. Somewhat simplistically, the way I think through this problem is that one needs to identify a target, that is a disease target, a cell target, a mechanism that one then tries to build a molecular intervention towards to intervene in that disease process. Often forgotten, although I don't think by the folks in this room, is that one then needs to deliver that package, that intervention to the appropriate target. And what we've seen over the past couple of years is that there's just an explosion in terms of the targets that we consider, and some of those are listed here, or some of those variances on that targets. We have new ways of molecular intervening at such targets. And one of the premises of my talk here is that given this maturing field, given the new abilities we have and the advanced questions we're asking, many of these modalities require really very specific gene delivery requirements, or need to address very specific gene delivery requirements. So in that, to some extent, this work has grown out of both this opportunity as well as a frustration. So we have been doing work on building vectors for gene therapy for, like I mentioned, short 20 years. So one would argue that there's some level of expertise that comes with that time in the field. Yet 6 years ago, when I opened up my lab, the questions that came to me from this growing and maturing field, I frankly couldn't answer. These are some of the questions that I was asked and are still being asked. Can I detarget liver? Or the next person could say, can I target liver more efficiently and selectively? Can I administer to a seropositive patient for serotype AAVX? Can I have a higher yield? And often these are in combination. One of these questions is, can I package a Cas9, a guide RNA, and a homologous recombination cassette in one vector preferably? And if you could give it a hit and run pharmacokinetic profile, that would be great as well. Even though we've been doing this for a long time as a field, these questions, there are no real direct ways of addressing these. And frankly, we often don't have the tools available or a methodology to get there. So what I'll try to walk through here is both philosophically as well as empirically how we are trying with baby steps to address these questions. We start off from an incredibly potent mold AAV, which I won't introduce to you because I think it's well known by many of the talks here. AAV as a starting point is an incredibly elegant, potent, safe tool for in vivo gene delivery and maybe with some opportunities in the ex vivo space as well. There's a few premises to the work that I'm doing. Some of them I've highlighted in a previous talk. But very briefly, we've learned over the years that small natural variation, as well as engineered variation, by the way, of AAV capsid composition can dramatically alter a plurality of viral phenotypes. And these viral phenotypes are often directly relevant to gene therapy translation. Some of these are listed here. They speak to crossing blood-brain barrier in the VEX's application, for example, for SMA. They speak to manufacturing, assembly, and packaging. They speak to immunogenicities. One little vignette, something that we've actually known for quite some time, and many in the field of AAV development have looked at AAV1 versus 6. These are serotypes we've had for a long time. They actually differ only by 6 amino acids. And through some of the work that we have published and others have published, there's this particular lysine residue that create both in the middle here on a biophysical level. Let's see if I can actually have a pointer here. On a biophysical level, at this angle, I'm actually going to give up on this this early in the morning with possibly a little bit too much alcohol yesterday. So on a biophysical level, these differ, and this one residue determines that biophysical shift. And on a functional level here, when looking, for example, at liver versus heart targeting, you get these phenotypic switches. Again, a single residue can cause this. Just imagine the kind of perturbations that people in the field are doing now with dozens of mutations, what the effects of that may be. Premise number 2 is that to disentangle these different AAV functions is incredibly difficult. And why is that? Because of the structure, which also we've had incredible resolution of by significant work on the structural side. And the structure really is a 60-mer that comes together, 60 building blocks come together along perfect and pristine axes of symmetries that are illustrated here in this icosahedron on the bottom right, that one needs to manage and contain each time you perturb this structure. The analogy that I have is that this is like solving a Rubik's Cube, where one tries to get one function, say all the orange fields lined up, but at the same time to maintain the structural integrity as well as some of the desirable biology features that you want to keep, such as say liver tropism, you need to line up all these colored fields, and you all know how difficult that is. Moreover, we're doing this in grayscale. We often don't quite know what determines these different colors or these different features. To give some more context to this, this is a slide made by Eric Sin in the lab. This is an illustration of these defining axes of symmetry. In green, the two-fold axis, in orange, the three-fold axis, and the five-fold axis in blue. And when you start outlining some of the network interactions of residues that are expected to have some kind of an electrostatic interaction, this is the spider web one gets. Again, a perturbation in the spider web needs to be compensated in a variety of ways, often without any real understanding of how to achieve that. So how's the field tackled this? Very creatively. We've used the tools that have come to our field and to the molecular biology field in general, starting with some of the efforts that I was part of through biomining, where we just went through nature and tried to find novel viruses. And effectively, this was incredibly productive, and some of the viruses like AAV8, AAV7, AAV9, and so forth that are now in the clinic came out of these efforts. People have also tried to engineer through directed evolution methods, and you've seen a lot of that at the meeting here. Somewhat less successful have been the rational design, again, because of the limitations of the understanding that we have on how to effectively integrate these perturbations that we want to do. Quick illustration on why I think some of these efforts have succeeded and sometimes have not succeeded to the level to address these questions. So this is what I call the solution space that we're working in, or actually the perturbation space, which is massive. It actually quantitatively could be all the way up to the power of 20 to the power of 740, which is the number of residues that the AAV capsid has. Each time we want to do a perturbation, we want to do that with a certain bias. But obviously we want to retain that overall functionality, and those are the two axes here highlighted. And we want to get to that solution space in the bottom right, which depending on the ask can be very, very small relative to this overall diversity space. So I would argue biomining efforts skew things towards the functionality axis or maximize the functionality axis, but are somewhat unbiased. And exactly that's what we found over the years. When we do a directed evolution effort, we basically take a very small sampling, even though these are often millions of variants. We take a very small sampling, and by the way, this is not drawn to scale, that somehow work towards bias and somehow work towards functionality, but are really kind of just a hand of sand that one takes out of the desert of possible options. And so therefore, it's somewhat, you have to be somewhat lucky to land in that interface between the solution space. And if the solution space is too small, just purely statistically, it becomes very difficult to find something that meets the criteria that you want. Rational design, again, arguably the least successful of the strategies that have been employed, is obviously much more biased, but it's very quickly that we lose functionality. And that is often seen that many of these variants that we generate are less functional, for example, in just sheer assembly function. So what is the path forward, in our view, towards improved rational design? Well, we would like to leverage more mechanism, and that means that we have to understand more mechanism. Related to that is that we have to understand the structure function. We don't just need the structure, we actually need the functional consequences of structural perturbations. And then we need targeted and efficient evaluation methods. One of the efforts that I can go into depth about is Amanda Dudek, who's a student in the lab, and who, by the way, is looking for a postdoc. And has had several very nice posters here, that is trying to get more of the natural biology established in terms of, for example, receptor entry and trafficking mechanisms. Another philosophical stance, and then I think I'll dive into some more data, is that we would like to address actually the most difficult questions first. And some of these most difficult questions are, how do we just build a stable particle, for starters? If we know the rules on how to build these particles, then maybe you can put the bells and whistles on later. One of the key issues for this field is, and I call it the Achilles heel of the AAV field, is neutralizing epitopes, or the problem of pre-existing immunity. And one of the goals that we've set forth a long time ago is, how do we ultimately move ourselves to developing a particle that is entirely inert towards neutralizing antibody, because it's a goal that none of us have achieved yet. So there was a naive idea that emerged probably about 5 years ago in the lab, and that is, to some extent, all these functional tests the virus has walked through just by passing through evolution. And many of the things that put pressure on this virus to evolve are these functions that we want to study, such as assembly, packaging, stability, species specificity, maybe transduction efficiency, and host response. And there was somewhat of a desire, can we rewind the evolutionary clock and somehow in the lab empirically interrogate or replay that evolutionary pathway. So that was the naive idea that I was incredibly fortunate that I had a technician at the time who, after he was pouring LB plates, devoted some of his time, and it became ultimately a full-time effort, to develop ways of us getting to that approach, again empirically. And very briefly, the way this worked, we've published this in 2015, is that we use the available data sets on sequence for AAVs. We put some filters on those. We built a robust phylogeny, which in and of itself is not easy, because this phylogeny ends up being the roadmap for us to get to these red dots, these nodes, where not only structures diverge, but also phenotypes diverge. And then we do a statistical prediction of what the sequence may have looked like. These are not genuine ancestors, they're approximations of what this ancestral state may have been. And we synthesize those in the lab, and we want to functionally evaluate those. I am very much an empiricist and not very much a modeler, so I was skeptical that this was going to work at all, even though I thought the idea was creative. So we hedged our bets. The way we did this is by building statistical spaces. We actually tried to take some of the ambiguities of these models into a statistical approach that we called this probabilistic ASR, or ancestral sequence reconstruction. That's illustrated in this blue diagram around the nodes. Basically what we wanted to do is we wanted to capture all of these potential variants or potential ancestors, if you will, in a library space, synthesize those library, and now interrogate that entire library. And the initial attempt was just to have at least one functional virus to study, because I was skeptical that we wouldn't have, out of this long effort, wouldn't have anything to study, because none of these particles would functionally assemble. So this is illustrated here. This is a trimer, and in blue are all the variations that are introduced compared to a contemporary or an extant serotype, like AAV8 in this case. And in red are those ambiguous residues where we couldn't quite call it. And what these residues really do is that they define, somewhat simplistically speaking, one branch in one state, kind of the branch going, say, north, versus a branch going south in another state. And again, we wanted to keep that ambiguity alive and interrogate exactly what these residues were responsible for. When we picked our most probable ancestor, we actually were surprised. There was one green cell. When we produced some virus, put it on cells for transduction, we found approximately one green cell per microscopic field. Not a great transduction efficiency, but we were anticipating nothing. So this was a success. And when we, in a somewhat pedestrian way, walked through all the variants, actually not even all the variants, but a significant amount of the variants in that blue space, we found that certain viruses were actually somewhat more effective, like ANC-ADL65 illustrated here. Let me introduce you this one validation, if you will, of this approach, which we call kind of a lead, and it was arguably somewhat arbitrarily chosen, but it ended up being a very interesting virus to work with. These viruses produce up to the level of AAV2. They biochemically and via transmission electron microscopy look very analogous to what we expect from an AAV, and we looked in a variety of biophysical ways as well. For all intents and purposes, this virus still very much resembles what an AAV should look like, and we're actually defining structures via cryo currently. These particles are divergent. We removed ourselves quite far from the space that we're currently working with in the clinic, and these are just some numbers and some illustrations on the capsid surface, and this speaks to epitopes relevant to neutralization. We diverged ourselves by roughly 20 to 30% compared to other AAVs that are circulating in humans. And this is one of the ambitions again, right? We want to develop something that is antigenically as distinct as possible. And at least we felt that we made a mark at some level here from a structural perspective. And then the big question was, are they still somewhat relevant in vivo? And the answer there initially was yes, seemingly so. We showed that for liver, for muscle, and for retina, we had basic transduction efficiencies, and actually quite often they were somewhat in line with the efficiencies of some of the, if you will, gold standards in the field, such as AAV8. We followed up with a whole series of publications listed here, which I won't go through in detail, But we looked at safety, we looked at liver, we looked at retina, CNS, cochlea, and a variety of other targets. And overall, this seemed to be a worthwhile vector to pursue for clinical applications. This is one illustration out of a collaboration with MGH, with Ava, Brad Hyman, Casey McGuire, and Eloise Hudry, where we wanted to, for example, compare this to AAV9 in an IV administration in mice, looking at blood-brain barrier passage. And indeed, when we look at the CNS, we see that in a single-stranded context, there's a quantitative benefit to using ANKD compared to AAV9, which, as you know, is currently in the clinic. Single-stranded AAV9 performance still outperforms ANKD. We are interested mainly in larger cassettes, and we're not exploring too much the single-stranded single-stranded technology. Our ambition is really to work with the full capacity of AAV, the 5 kb. So, but what are we learning about the structure function? We've developed a few more tools based on this initial success. One of these tools is that we can now actually do a full lineage reconstruction, where we can narrow down in smaller steps the genetic variation across multiple nodes. So all these red dots we have brought to life in the lab, and again, one can walk through and see different residues coming into play. And if we now just have an ability to interrogate the function of each of these residues, we think we may get closer to a structure function definition, at least of the residues studied. And indeed, this proved relevant, because if you walk through some of these clones, you can look, for example, at the structural determinants of assembly, and you see that there's stepwise increases along that phylogeny. I think in the interest of time, and I covered this in a previous presentation, I'll just very briefly go through kind of a vignette of how we can go even more in depth and start looking at some mechanism. And this requires some knowledge about AAP, which is an assembly factor that is known to be essential for many of the AAVs to basically form itself. It's the glue that holds AAV together, or it's the glue that's necessary when building a particle. This is illustrated here when you basically put a stop code on an AAP, that for AAV3 and AAV9, one has orders of magnitude reduction of titers. Anna in the lab explored the function of a specific conserved domain in AAP, and if you move up the stop codon to past this domain, one sees that one can actually rescue AAP function for many of the serotypes that otherwise are dependent on AAP. And it's this particular domain between 20 and 40 that is relevant. We can now overlay these phenotypes, this assay, on a phylogenetic tree, and in red you can see AAP dependency, and in green what we call AAP-C independencies. And now one can go to these junctions where red turns into green to interrogate how AAP interacts with that capsid. And we call it branch D, branch I. We look at the conserved residues in D versus I, and we came out to 11 residues, exactly 11 residues, that when you look at it structurally, you can see that these 11 residues in the independent states have particular bonds, salt bridges, for example, in this example, whereas in the dependent states, those bonds are absent. So basically, it seems like some of the glue sometimes can be provided by the capsid itself, and sometimes it needs that additional glue. In addition, when we transfer these 11 residues, we can transfer that independence, meaning in terms of rational design, we can, if this were to be a pursuit, we can transfer this particular property from one virus to another. And the methodology through this phylogeny to phenotype mapping, or vice versa actually, allows us to do that in a very directed manner, because it's exactly these 11 residues that are, by the way, illustrated here in green. These are not linear domains. These are epistatic networks that are required to achieve this. Each of these residues was essential to this function because we looked at every single one independently and in combination. And illustrated here on the bottom right is, again, that increase that is achieved when you transfer those residues. So we want to use this type of a vignette, but do this at a larger scale for more complex functions, actually, even though AAP in and of itself is fairly complex. What we've done is we've taken this approach of probabilistic space, overlaid it to all of these nodes, built all of these types of libraries, which leads to about 40,000 different sequences that at some level interrogate this entire evolutionary pathway. Now, the next question came up in one of the first slides. How can we functionally interrogate those complex functions that we're interested in for gene therapy development? And do this effectively in a screen that actually exhaustively captures quantitatively all of these functions. Barcoding, you've heard much about at this meeting. We've incorporated it into our work, and this is illustrated here, and basically the basic principle is that one now starts to characterize the input, the output of a particular selective screen, and you look at enrichment of certain species or de-enrichment of certain species. Before we even went there, we felt this methodology is rife with potential artifacts, and we did what we call due diligence, and Pauline Smith took a senior role in that in the lab to just look at things like reproducibility, proportionality. AAP actually is a concern in trying to achieve this. We want to build some internal controls, look at things like cross-packaging and mosaicism. Just a few elements of that work. In terms of the quantitative nature of the assay, we can actually skew libraries one way or another and then monitor if our NGS readouts allows us to monitor these quantitative known effects that we had put into these test libraries, and the answer there is yes. We can see this both on a plasmid level as well as on a viral level. Simon earlier this meeting presented on this nagging issue of mosaicism. Mosaicism is when you do produce a multiplexed library, one can get these hybrid particles that are illustrated on the bottom here. We've developed previously, based on work from Mavis McKenna, this assay that we call AAV-ID, which allows us to monitor for these mosaics. On the top here are certain conditions where you see these peaks in the middle. That actually shows you that there's 4 intermediate species which illustrate that there's mosaicism. We can bring down conditions to a level that these mosaics are minimized and likely not going to provide too much of a noise function in our work. How reproducible is our work? We don't cycle like people do in directed evolution. We look for reproducibility along replicates. This is basically a correlation, or it's a Spearman's R that shows that 0.99% reproducibility when we look just at the method itself in terms of redoing the same experiment twice. Things like competitive interference are also relevant when you run multiplex libraries, and those things we test as well by basically looking when we have high competition versus low competition, do we make the same observations? And the answer there overall is yes, but it also illustrates that we need to meet certain levels of sensitivity, and at low dose, we're getting closer to that sensitivity line. Now, what we can actually do after all of this due diligence with this method is illustrated here. We put in a certain amount of plasmid, and we look at the assembly function. Can we make these particles? What is illustrated on the right are particles that are enriched versus de-enriched when looking at one of these complex libraries. And the nice thing about this is that we can immediately, with our barcodes, break this down to the functional determinants of that function. There's about 100-fold spread in the variance that we just saw between the most performing or the best assembling virus and the least assembling virus. Some of these red dots on this capsid do not play a role in this assembly function. Some of them do. Some of them skew things one way or another, as is illustrated here on the right. But then we bring it to a more complex physiological system like an IV administration of a mouse, but there's actually nothing that stops us, and there's nothing that's stopping us to do this, for example, in a complex model like a non-human primate. Here we see a divergence of 2 populations, which is quite interesting, towards a particular tissue tropism. Again, we can overlay this with these particular residues that we are toggling. Some play no role, some skew, and we actually start seeing a multi-parameter function that we get increased packaging and increased tropism on the top right. And then some of these residues track exclusively with this particular tissue tropism, as is illustrated here. We think that the data coming from this methodology gives us and is giving us an incredibly big data set, yet it is still sparse in terms of understanding the landscape function on the structure function. And eventually we need methods like machine learning to kind of fill in the blanks. So with that, I want to close. I think I've done my acknowledgements throughout the slides. We think we're on a trajectory towards moving viral vector systems that we've adopted from nature to synthetic particles that we can reprogram eventually. This is a slow process. We're nowhere close to having a final to addressing all of the questions that I put in one of those first slides. But I think with these types of steps and those of others in the field, we're getting there. So with that, if there's still time, I'm happy to take some questions. Thank you.
So when you look at the tissue tropism, what tissues do you look at? Because is there a specific tissue you pay more attention to, or you just look throughout the entire animal?
So we have specific tissues for our translational efforts, right? These questions that I put up front are questions that are coming for our personal translational efforts, but also because of the industry partners as well as the academic partners. And because of that, and because of the potency of the assay we've developed, we actually harvest as many tissues as we can. Most of our mouse studies are 12 tissues, and the data we're getting back is quantitative data, for each of these tissues, that we can also look at things like selectivity, right? What goes well to the liver, but doesn't go well to your little toe. And you can select the optimal across these multiple parameters, and I think that's one of the advantages of what we've developed.
Your phylogenetic tree has a few nodes left to go. Is your ancestral modeling capable of going farther out and possibly getting an unmutated common ancestor for all AAVs?
Yeah, so as you probably noticed, we're trying to approach this as quantitative as possible, and we're interested in exhaustively capturing the variation we define. And the problem with these older nodes, at least in our initial efforts, is that there's too much ambiguity to handle. When there's too much ambiguity, you go into issues like subsampling the space that you create. So we've held off on that, and our approach is actually to do this in an iterative process. The lessons learned from what we are gathering now allow us to refine our models to go back exactly further down that phylogenetic tree.
Can I follow up on that, Luke? Because it's a really good question, because when we used ASR for factor 8, we were skeptical as well on trying to, the nodes that we looked at and made proteins for, but they were all active, and we were very surprised that you could go back into these trees and get an active biologic with the mutations, the number of mutations that were made. So have you seen the same thing where each of these nodes have are actually functional, or have you found non-functional variants?
We've actually found functionality, and it depends also on the sensitivity of the measurements, in a surprising amount of the variants that we identified. Estimates are, depending again on the sensitivity, between 60 and over 80% of the variants. That being said, for example, there's one particular node that we consider a DUT, where assembly function is just down. But overall, the 8 other nodes that we have developed seem to be pretty potent and promising, sometimes even leading to assembly functions that are higher than some of our most performing viruses. But you're right, I mean, I think we're basically leveraging kind of evolutionary power to narrow down the variation we're looking at, which biases towards functionality.
Hi, Yuan Lu from Wuxia Tech. It's really a good talk. I'm wondering, compared to the previous direct evolution, that you mentioned that you're no longer using continuous runoff selection, and you use repeated and see the same reproducibility. What's the rationale for that?
The rationale for that is, I mean, like we do repeat experiments in biology, right? We do the same experiment X number of times, and we only report on those things that are consistent observations across. The reason why in directed evolution people passage is to work around issues of sensitivities, which we actually are fortunate not to have. The sensitivity of the NGS readouts allows us in each of our rounds, actually there are still issues of sensitivity, but in some of the situations that we look at, we have an exhaustive readout on each of the variants in this pool. And now we do this experiment the second time, and we're only going to accept those things that have the same observation in one mouse versus another, or one monkey versus another, for example. So we don't need to do the passaging because the sensitivity issue is reduced in our context.
So if you increase the sensitivity, were you doing a continuous for an enrichment, or you will still use the reproducibility method you are using?
No, we don't do enrichments. The levers we have on sensitivity is the dose that we inject, is a clear lever. We inject more, we get more sensitivity. and the other lever is just the power of NGS. And I have to say we use PCR to amplify the barcodes to get to that readout. So it's basically leveraging the sensitivity of PCR and NGS to get us that sensitivity.
Thank you.
Thank you. Okay. So
Thank you.
Thank you.